Lysosome-related organelles, such as pigment cell melanosomes and platelet-dense granules, are a class of cell-type-specific subcellular compartments. Defects in the trafficking pathways that form lysosome-related organelles cause Hermansky-Pudlak syndrome, a disease characterized by albinism, excessive bleeding, and other symptoms. Michael Marks from the University of Pennsylvania explains that the genes mutated in Hermansky-Pudlak syndrome encode subunits of several protein complexes that help transport cargo from early endosomes to lysosome-related organelles.
We've been trying to understand what these protein complexes actually do during the formation of lysosome-related organelles. So we had shown before that BLOC'd is required for the exit of most of the cargoes of melanosomes, at least, out of early endosomes towards maturing melanosomes. And there's another complex, AP3, that's mutagenized in Hermansky-Pudlak syndrome type 2 patients. And we and others had shown that AP3 regulates the trafficking of tyrosinase through a BLOC'd independent pathway, also from endosomes towards melanosomes. And so next on the list was BLOC'd.
The BLOC'd complex is composed of 3 subunits encoded by genes mutated in Hermansky-Pudlak syndrome. Marks and colleagues, including Megan Dennis, Adriana Mantegazza, and Subarao Gangisetti, found that melanocytes derived from BLOC'd deficient mice showed reduced levels of melanosome maturation and pigmentation, a phenotype rescued by the restoration of BLOC'd function. The BLOC'd independent cargo tyrosinase was, for the most part, still delivered to peripheral melanosomes in the absence of the BLOC'd complex. That wasn't the case, however, for BLOC'd dependent cargoes such as tyrosinase-related protein 1.
The proteins that are part of this BLOC'd dependent pathway seem to be all over the place. So there was a little bit of stuff that was trapped in endosomes, like in the BLOC'd mutants. There was an increase on the plasma membrane, which we also see in BLOC'd mutants, but it wasn't nearly as bad. And the weirdest thing was that there was this huge accumulation of stuff in the Golgi complex.
BLOC'd localizes to recycling endosomal tubules that deliver cargoes such as tyrosinase-related protein 1 to melanosomes. Dennis et al. Reasoned that, in the absence of BLOC'd, these transport intermediates might be delivered to the Golgi and plasma membrane instead, an idea they tested by monitoring the fluorescence recovery after photobleaching of GFP-tagged tyrosinase-related protein 1.
So you get very little recovery in the Golgi region. It's in a wild-type melanocyte. But if you do the same experiment in the BLOC'd mutants, you get this very rapid recovery of material from the Golgi. And if you look at it by imaging, you can just sort of see this movement of vesicles towards the Golgi region from the periphery, consistent with this idea that in the BLOC'd mutants, you actually have an increase in retrograde trafficking of these cargoes back to the Golgi complex.
Endocytic recycling to the plasma membrane was also increased in BLOC'd deficient cells, explaining the increased levels of melanosomal proteins at the cell surface. To learn more about how BLOC'd might target recycling endosomal tubules to melanosomes, Dennis et al. Followed the dynamics of these transport intermediates by live cell imaging.
So in wild-type cells, you could see these beautiful elongated tubules that harbor recycling endosomal markers like syntaxin 13 or Rab11 that emerge out of endosomes and can transiently wrap around melanosomes. Some of them don't hang on for very long, but a good fraction of them last for like more than 20 seconds. You can see some of them lasting for up to a minute, contacting these melanosomes. In the mutant, you just don't see that. Almost all of these tubes are really, really fast, and if they contact a melanosome at all, they let go within a few seconds. So it looks like the ability to reach the melanosomes and to hang on to those melanosomes once they do reach them is compromised in these mutants.
BLOC'd and BLOC'd therefore control the transport of the same cargoes from endosomes to melanosomes, but appear to operate at different stages of the pathway.
So BLOC'd is required for basically getting the cargo out into these tubes. And then the most likely model is that block 2 is acting as a tethering complex to basically tether these guys onto the melanosomes. The other possibility is that it simply stabilizes the tubules in some way so that they have time to find the right target and make those contacts. We're now trying to purify recombinant block to it, and the prediction, if it's a tether, is that it'll interact with the snare proteins that are involved in this process. And so that's the thing we're trying to figure out.
In the meantime, however, you can learn more about how Block 2 targets recycling endosomal tubules to melanosomes for cargo transport in the paper by Dennis et al., published in the May 25, 2015 issue of the Journal of Cell Biology.
You